So in this session, we're going to look at the study designs in nutritional epidemiology. The key issue that we are interested in is the relationship between diet and health outcomes. The food that we eat over time, how does it affect our health, and what diseases actually happen? So for us to understand the answers to those questions, we need to carry out studies or research. So we need to use the appropriate research designs to be able to come up with the right answers. So we have a variety of research designs that we can use to generate answers, or basically to generate evidence for the existence of a relationship between diet and the health outcome of interest. So generally, we can say that we have two broad categories of research designs: the interventional research designs and then the observational research design. So in this particular case, it is very often that we carry out observational research designs. With observational research designs, we look at the individuals that we are actually studying, or the participants in their natural setting. We don't intervene. Okay, we don't intervene. So we can call these studies non-experimental or non-interventional studies. Whereas the other broad category is what we call experimental or interventional studies. So with the observational studies, we are basically interested in knowing the association between the dietary exposure and health outcomes. We just need to know that relationship, that relationship exists, and if it exists, it is in which direction. But it does not actually tell us. Observational studies do not tell us whether a particular dietary exposure is the cause for a particular health outcome. Just tells us the association. So observational studies can be cross-sectional studies, cohort studies, or case control studies. And we are going to look at each of these. Cross-sectional studies, with this, you just go to a particular population and then carry out an assessment or collect data at that particular moment. So usually this could be what we call a snapshot study, where you just get what is existing at that particular moment in time. So we may be interested in knowing how many people have hypertension. That is a health outcome. Or how many people consume a lot of sugars. Okay, we basically want to know the prevalence. Yes, and that's all. That's what cross-sectional studies will actually tell us. Cross-sectional studies are very cheap to carry out. They're effective. You can generate an understanding of about the occurrence of a particular risk factor. So you generate what we call a hypothesis, or you can generate hypotheses. This one will provide you with some information about the prevalence or the occurrence, or you get the pattern of occurrence of a particular disease. So that's the benefit with the cross-sectional studies. The issue is they do not tell us the cause. They don't tell us more about the cause, or what we call the cause and effect relationship. You will just find out that, okay, you have gone to a particular population, you want to find out how the extent of occurrence of hypertension. And then maybe in your questionnaire, you have questions related to the risk factors of hypertension. Okay, you'll get the quantities of whatever parameter that you're interested in, but they will not tell you that this parameter is like this, it is done causing this. It does not give you what you call causality. But still, you will get issues to do with biases. So those are some of the challenges that you get. Biases are when you don't get accurate results. This could be because of interpretation of the research questions, or maybe the data is not collected very well. And in this particular case, you may not really get the long-term data rate pattern of individuals like that. You can establish a relationship between the health outcome and the data exposure. So that would be one of the setbacks of cross-sectional studies. Then cohort studies is when you follow people over a period of time to assess exposures and outcomes. So for example, if people maybe take a particular type of food, and then you study them over a period of time and then see who develops a particular disease and who doesn't. That's what we call a cohort. You basically follow people over a period of time to understand if they will develop a disease of interest or not, based on their dietary history. You can carry out cohort studies in two ways. You can look at people going forward, that is, you study them for the next 2 years or 1 year or 5 years. The next, okay, that is prospective. Retrospective is you collect data, their past dietary habits, or you collect data about their past dietary habits. You look at information and then look at their outcome. So basically, you start with the outcome, whether someone has the outcome of interest or not, and then look at their dietary history. Okay, so that is retrospective. And then we look at the advantages and disadvantages. Of course, they can tell you about the existence of a temporal relationship compared to cross-sectional. They tell you more compared to cross-sectional studies. And then you can investigate multiple outcomes and maybe establish a relationship between the outcomes and the exposures. The fact that you investigate this relationship over a long period of time means that the research may actually be expensive. So sometimes the people that you actually study may drop out of the study, and then you get problems, biases, because of loss of follow-up. But measurement errors can also happen, and also the effects of confounding factors, which we explained earlier on. So the other category are what we call the case control. With this, you compare people who have a particular disease of interest, who we call cases, and compare them with those who do not have the disease of interest, and then look at their past history. So you can be able to get information about what could have been the risk factor. Okay, especially if the conditions are actually very rare. So we conduct case controls when the conditions are actually very rare. So the advantage is cost-effective, quick to do, but still, you may get challenges on the fact that some people may not recall their past exposures. So, for example, you get a person with a particular condition, you ask them questions, and then they may not really be able to give you the accurate answer to those questions. So that is one of the challenges of case control studies. Okay, they can help you with establishing the relationship, the temporal relationship, but it can quite be difficult because of issues related to biases and other measurement errors. So we have what we call ecological studies. So in this we look at populations. The key difference is that we look at populations. In all the other studies, you have looked at group, individuals. Okay, maybe an individual who takes this type of food, will they develop this disease? Okay, but in this case, with ecological studies, we look at a population level. Okay, we are at the population level. So, for example, we look at data from a given state and say how many people have hypertension in this state. So we basically look at the data that is already collected, and then we compare that data with another state, perhaps. Okay, and then be able to make conclusions based on the populations that we are actually investigating. So, if, for example, people in a state X have, let us say, they consume a lot of salt, and then also the number of people with hypertension in that state is also very high. So we can be able to draw a conclusion that there is a relationship between high salt consumption and hypertension. So that is what we call an ecological study. We look at data which is already collected about a particular population and then be able to draw a conclusion between health outcomes and exposures. In this case, we're specifically looking at what you call dietary exposures. So when do we use, or what are the uses of ecological studies? They generate what we call hypotheses. They give us a first step of understanding whether there is a relationship. Okay, and then we can investigate that relationship further. So it helps us generating what? Hypotheses. And then the next one is that we can get the general pattern of a particular condition. In this case, the general pattern of dietary consumption and how they can impact population, just like the example of salt consumption and hypertension. We can therefore use actually ecological studies also to inform our decisions in the resource allocation and what programs we can actually institute. Before we institute a program, we can come out an ecological study and tells us something, and then we can implement a project. So what are the advantages? Of course, it's more easier to do. You use already collected data. You don't have a lot of resources to use, and it can be easy to conduct in a timely way. You get hypotheses which you can investigate further, and tells us a lot of information about dietary patterns. So we basically use the fact that we basically have the advantage of using information that is already existing. I see the benefit. The other one is it can help. Ecological studies can help in designing public health policies and interventions. The challenge is that you get what you call ecological fallacy, which is that, you see, the data that we are looking at is based on a population. That may not be true for individuals. Okay? If you have an association at a population level, that association may not apply at individual level. That is what we call ecological fallacy. Okay, the other issue is what we call ecological bias, that you will get wrong interpretations based on biases as a result in the ecological studies. The biases happen because of one confounding factors that have not been accounted for during the period of analysis. Remember that confounding factors are those that influence the outcome of interest, and you need to take care of them, okay, in your analysis and also generally in your research study. So the other issue is that you rely on existing data. That existing data may have issues with quality, reliability, and sometimes you don't actually have it. So that's one of the problems. And then when you have population data, that does not necessarily mean that you'll actually have individual level data. So that's also the other problem. Then the other broad category of research designs are the ones we call the experimental. We can call them interventional studies also. With this, the researcher actually manipulates the exposure and then assesses the outcome under control the conditions. The most common types of experimental study are what we call the randomized control studies or randomized control trials. They give us the best evidence for establishing whether whether there is causality, whether a given risk factor is the cause for a particular health outcome. So that is randomized control trials. But we can also carry out what we call community trials. Okay. With community trials, for example, you can have an intervention at community level and see what impact will it have compared to the community that does not have the impact, the intervention of interest. Okay. So, for example, if you add fluorine to water, to a water source of the given community, and then you don't have fluorine added to water in another community, so you look at the health outcomes in those two communities. When you carry out something like that, that is what we call a community trial. So the advantages are that they give you the best kind of evidence that you want, because it can establish causality, which we actually lack in most of the observational studies. Okay, so you basically directly measure the health outcome of interest, and you have good quality evidence. The challenge is that it is time-consuming and expensive. You can still have some limited ability to generalize your findings, and then it requires a lot of ethical approval. So therefore, compliance with the ethical issues becomes a challenge. The last type of design that we have is what we call meta-analysis. With this, you basically look at the different research studies that have been conducted, and then review them properly, select them rigorously, and then make a synthesis, synthesize all the findings from different studies, and then come up with conclusions. Okay, so basically you look at what other people have found in their studies, and then make new conclusions based on what you have found. That's what we call meta-analysis. So how do you use that? For example, one of the challenges of that we have in nutrition epidemiology is that, you see, there are many factors that contribute to the occurrence of a given disease. Okay, for example, we say diabetes can have genetic predisposition, or it can have issues to do with one's lifestyle or nutritional habits, dietary habits. So how are you going to be able to say that it is indeed true? So you may not base on only one study. You may need to have a variety of studies for you to reach those conclusions. So when you carry out a meta-analysis, it means you're going to look at all the studies that have investigated the occurrence of diabetes. And then by looking at all these different studies, you'll be able to say, yes, diabetes is because of consuming a lot of sugary foods. Okay, so it helps you in summarizing the evidence that you have. But also, For you to decide on which study you're going to look at, you need to have established a parameter. It means you are going to look at the different studies, have those that you will accept, have those that you will reject, say, no, I will not look at this document because the research was not done in the appropriate way. And so when you conduct a meta-analysis, it means that you are looking at the issues to do with sample sizes, and sample sizes actually have a big role to play in what we call statistical power. So you have evidence that you actually can believe because of the statistical power that the different studies actually have. And then also the occurrence of a particular health outcome may be influenced by a number of factors, depending on the populations that you're actually investigating. So populations are not the same. If they are not the same, we say that they are what we call, there is what we call heterogeneity. Okay, they are not what we can call homogeneous. They are not homogeneous. They are heterogeneous, meaning that they are actually different. Populations are not the same. But if populations are not the same, but actually this is most likely what is going to happen in a meta-analysis. The populations are heterogeneous, they are different. But if you come up with similar conclusions, then there is actually strong evidence that you're gathering. Okay, so it also tells you about the fact that other factors could be at play because populations are not the same. So that's one of the important things with meta-analysis. Sometimes we have conflicting ideas, and in nutrition it's very common. Someone tells you don't take wine, and then another nutritionist will tell you please take wine. So you have conflicting recommendations. So how do you solve that problem? You look at the different studies that have been done about wine and health outcomes. After looking at every study that falls within the category that you want, you come up with good evidence and say, yes, wine is not bad if you take this kind of quantity. So meta-analysis can help you in that situation. So how do you carry out a meta-analysis? First, you need to have a problem that you really want to solve. So you need to have a research question. Is it that you want to identify the relationship between wine and health outcomes? If that is the question, then you write that research question. And then go and carry out research. Go and look at the literature. Look at the publicized studies. And then select the studies that you think meet the criteria that we have set. Because you don't pick any study and then say, according to this study, this is what they said. No, you need to look at this study reached at this conclusion. How did they reach at this conclusion? How did they do the research? Who did they interview? What are the sources of bias? You have all these questions that you need to answer before you include that study into your meta-analysis. Okay, so you select the studies, the ones that you think can meet the terms that you have set, and then start getting the data from the different studies. You may create a table. Create a table where you have, let us say, study participants, their characteristics, the study type, what was the design, what were they investigating, what did they do? Okay, how did they measure what they measured? How did they even conduct the data analysis? Okay, so that is data extraction. And then now you can analyze the data from the different studies using the appropriate statistical methods. Okay, and then now you can draw conclusions about what you have got. Okay, so the advantages, it is a systematic way of investigating a problem that you have. Because multiple studies are involved, you have high quality evidence. It is actually transparent because you have to document everything that you have done to reach at the conclusions that you have. So it is very comprehensive approach to investigating the problem. The challenges are that, yes, you have all these studies, but they may be actually different in terms of how they were carried out, the sample sizes, the analysis. They're not usually the same. But you could be interested in studies that have been published, but also know that there are some good studies that have not been published. And then you reach conclusion without having read about them. That's what we call publication bias. The other issue is that we have issues to do with data limitations. Okay, the information or the data that we have from these different studies may actually be incomplete, and you may not really get accurate and reliable data at the end of it all. Still, the effect of ecological fallacy can happen. Remember, we had ecological fallacy and ecological studies. Generally, and this is a weakness of nutrition epidemiology, we have errors in measurement. Okay, we can do all these designs, but there will still be errors. And that means that we'll get challenges making the right conclusions. We'll have the effect of confounding factors, which I've explained several times. People may not tell us the exact information that we may want. Maybe they forget. Maybe also the people that we selected into the study are not the appropriate ones. So we have a lot of problems. How do you choose the research study design? Depends on the question that you have. What resources do you have? What time do you have? Have you gotten the ethical clearance? Does the study pose a threat or a harm to the, does it pose harm to the participants? What are the potential sources of bias in that study? So in summary, we are basically saying that in nutritional epidemiological designs, we are interested in answering specific questions that we have that link dietary exposures with particular health outcomes. We want to investigate. We want to have evidence that when you eat this, it leads to this. So we can carry out research. So the researches, we have to be based on designs. And we have two broad categories of designs, the experimental and then the non-experimental. The experimental ones actually give us very good evidence compared to the non-experimental. So we looked at the non-experimental ones, the one we call the observational, where we looked at the cross-sectional, the case control, and then the cohort studies. We looked at the ecological studies, and then we looked at the interventional, and then the randomized control trials, and then the community trials, and the disadvantages associated with each of those, and generally that nutrition epidemiology usually has limitations as a field, that it is very hard to establish causality because of many factors. There are issues with measurements, confounding factors, and many other things. So that's the end of this presentation, unless you have any questions.